By burning fossil fuels, producing cement and damaging natural ecosystems, humans have been rapidly and dramatically increasing the concentration of CO2 in our atmosphere. In fact, CO2 levels are higher than during our entire history and are continuing to rise at an alarming rate. And this rising CO2 is the main driver of climate change, driving the rising temperatures that lead to disastrous extreme weather events becoming more intense and frequent, acidifying the oceans and damaging croplands and ecosystems globally. I'm Jasmine.
And I'm Chris.
And we're researchers from the University of Sheffield looking at how to solve environmental issues around the world. We're going to show you how climate scientists use lab techniques and fieldwork to study these environmental issues to help understand them and solve them.
So plants and soils might provide a solution to the problem of excess CO2. Plants and soils take up a good chunk of the CO2 we pump into the atmosphere, approximately a third of it, making them carbon sinks. This slows the rate at which CO2 builds up and consequently slows global warming.
As CO2 is essentially plant food, the more of it there is, the more plants can photosynthesize and the more CO2 in turn they can remove. However, plants may only be able to absorb additional CO2 if they're not limited by the availability of nutrients such as nitrogen and phosphorus, which play an important role in plant physiology.
So we're at the Bradfield Environmental Laboratory in the Peak District National Park as part of the University of Sheffield facilities. So we know that plants absorb CO2 from the atmosphere and pump it into the soil and slow the rate of global warming. But this rate of CO2 uptake is likely to be dependent on nutrient availability in the soil, particularly of nutrients such as nitrogen and phosphorus. And so we are testing a hypothesis here that nutrient limitation in the soil, particularly of phosphorus, might restrict a plant's ability to take up CO2 from the atmosphere. So here we have an experiment on grassland ecosystems, which are really easy to work with because we can just dig out chunks of them and move them to wherever is appropriate, such as here. And so we have extracted mesocosms, which are little cubes of intact grassland ecosystems, from a long-term experiment in Wardlow, which is also in the Peak District. And what this does is this creates multiple different states of nutrient limitation that we can then test to see how they respond differently to elevated CO2. So the experimental setup we have here is, first of all, we have two grasslands. One's acidic and one's limestone. And then we have 4 nutrient treatments. So we have a control treatment, which is just distilled water, and so we use that to compare against for the other nutrient treatments. We have 2 sets, nitrogen treatment, a low and a high nitrogen. And what these nitrogen treatments do is, through their interactions with other nutrients in the soil, is they manipulate the level of phosphorus limitation. And so the more nitrogen we add, the more phosphorus the plant therefore then demands, and therefore we can increase the limiting status of phosphorus. And then finally, we have a phosphorus treatment, and what that does is that eliminates any pre-existing phosphorus limitation from these grasslands. So in addition to the nutrient treatments, we also have CO2 treatments. And so this ring here is a mini face ring, and it releases CO2 over these grasslands. And so we're looking at an increase in the concentration of CO2 available to these plants by about 50%, which is 600 parts per million, which is a rate that we might expect to reach in the atmosphere toward the end of the century. And then we have a control CO2 group as well, which you can see are just these plots without the rings around them. And so this is just to compare the effects of CO2 between them. And so each of these grassland, nutrient and CO2 treatment combinations, we have 5 replicates of each. And so this helps with our statistical analyses, and it also gives us a really wide range of different conditions of plant and soils that we can get a good idea of what's going on under these treatment combinations. And this gives us 80 experimental grasslands in total. So one of the things that we wanted to understand here was, how does the rate of CO2 uptake vary under these different nutrient conditions? So that's what this robot here, called Skyline, this one's called Dylan, that's what this aims to measure. So this flux chamber, as you'll see, it might drop onto one of these mesocosms in a minute. So this measures the rate at which CO2 is exchanged between the plants, the soils and the atmosphere, and that can tell us how effective these are going to be at being carbon sinks. So one of the first things that I wanted to look at for my PhD research was, how has the community composition, which is the distribution of different species and how their abundance changes with treatment, how has this changed with elevated CO2 and nutrient treatments? So, to determine the community composition of one of these mesocosms, we use one of these. This is a quadrat. So, we place this on the corner here, and we can move all the vegetation through so it's not squashed. And then we can go through square by square, identifying the species present and estimating at what percentage of this square that they cover. And then when we record all of this data, kind of clean it up a little bit, we have an idea of the community composition of different plant species across this entire mesocosm. And so this is important because we want to understand how these different plants respond to elevated CO2 and nutrient treatment. So different plant groups or plant functional types have different ways of acquiring nutrients from the soil. So for instance, plants might invest additional carbon from elevated CO2 into symbiotic fungi, which help them get nutrients from the soil, or they might release carbon-based acids into the soil to mine phosphorus. And all these different ways of acquiring nutrients might affect carbon and nutrient cycle in the soil, and so it's important to understand how it changes. So to do this, we identify species from particular functional types that we're interested in, we sample them, so I can remove a lot of the material here, and then we can put them into some envelopes, take them to the lab to be dried and processed later.
So plants are really important for climate change because they're able to take in CO2 from the atmosphere, which is a greenhouse gas. But plants have a very special relationship with the soil that they're on. They're able to add carbon to that soil through their dead tissue, so when leaves fall on the ground or like dead plants decompose, but they're able to add carbon to the soil when they're alive through root exudates. So the CO2 they take in from the atmosphere, they convert it to sugars, and then that can be passed through their roots into the soil. And soil microorganisms, they control then how much CO2 is emitted from the soil. So soil carbon is considered a carbon sink because it's a place where we can store lots of carbon from the atmosphere. But carbon can take hundreds to thousands of years to build up in the soil, but it can be easily lost in 5 to tens of years. So we really need to understand what controls the soil carbon being released back out to the atmosphere through the microorganisms. To find out what's going on in the soil, we take soil cores and then we take the soil back to the lab. So this is a soil corer. This is quite a big one that we use. And then I use a mallet as well. We find a good spot and then I can mallet it in. Then we sort of twist it to dislodge what's there and pull up. And then we get a nice core of the soil. So it allows you to take sort of very good replicates of the soil, and you also get the nice organic horizon where all the roots are, and that's like the hot spot of microbial activity. And then you can just push the core out. You've got a nice little core. And now we need to go back to lab to analyse these samples. So this is one of the labs in the University of Sheffield. This is a drying oven, and we use these to dry plant and soil material. And then we take the dried material and then we grind it up. So this is some dry soil, so we can just pop this in. Put a little guard on. And then grind it up. As you can see, it's all ground up. So we're going to weigh about 50 milligrams of the dry soil that we just ground. So in soil, there's organic carbon and inorganic carbon. And we're only really interested in the organic carbon because that's what's available to plants and microbes. To remove the inorganic carbon, which is things like carbonates, which are found in like rocks, we use concentrated 6 molar hydrochloric acid. And this will react with the carbonates in the soil to produce CO2. And then we leave it overnight, so any of the carbonates can react with the acid and produce CO2. And then we leave it to dry, and then it's all removed. And we're going to weigh a tiny amount into this little tin boat. We only want about 20 milligrams. And then we have to take it, and we have to fold it up. So we can take our samples and we can run them on either a mass spec or we can run them on a CN analyzer. So the mass spec will give us a bit more information. It will tell us what type of molecules are in there, what type of bonds, whereas the CN analyzer will tell us the ratio of carbon to nitrogen in the sample. So this is the output from the CN analyzer. So you have your CN ratios, and I'll take this data and I'll input it into this software called R, and I use R to do statistics on the data to check that if we've seen any patterns or observations that they're statistically significant. So in this case, the data is showing that there is a significant difference between the two plant and the two soil samples. So in the high nitrogen treatment, there's a significantly larger amount of nitrogen in the plants in the high nitrogen treatment. So hopefully that's shown you a bit about the work that we do here and how we can use different techniques to study the environment.
So this kind of research helps us fight climate change because it's really important that we understand how environmental conditions such as nutrient availability impact the carbon cycle, particularly when we're creating changes in both the carbon and the nutrient cycle. And if we can understand how terrestrial ecosystems might respond in the future, we can understand how we might be able to mitigate climate change.
So I think the thing when we think about climate change is we think about saving nature, but really we need to save ourselves, and hopefully this sort of research will help us understand how climate change is going to change our environment and affect our food systems and our access to clean water and our access to green spaces.